grandfather and grandmother or uncle, they crossed more than 60 or 65, definitely they have some age-related diseases. Age-related diseases, the reason because their signature is signature in this sense, the DNA got mutated, and then that particular thing, is coming to the next generation, or it is late onset. Understand? That's what people are suffering. For that, next slide. Okay, so now I put nobody in the previous talk, nobody asked any question. So that's what I'm telling. Take your pad and pencil. Whatever you thinking in your mind related to this particular talk, or concerned with your research career, what I'm going to do, what kind of path I supposed to take, something like that. No, you write it in the notebook. You ask right now, or we will discuss during the lunch time. Okay, so better to ask a question. Without asking question, I think, see, if they brought up like in US, the baby, they never suppress the baby. She is supposed to ask why I have to do it, what, all the question, they will, even it's a kind of culture from the childhood. But the environment here in Indian scenario is different. If you ask a question, they're suppressing their individuality. So don't do that. So concerned with this particular topic, please ask a meaningful question. If it is a stupid question, I'm not going to answer anything. Ask a meaningful question related to this particular topic. You may have the problem, or your family having some problem. Sir, this is the problem. This is a genetic effect. What is the cause? Can you able to stop it, or is there any treatment? You can ask a question like that. Okay, so it means if you understand your own genome, if you know your entire genome, you can lead your life healthily till like 70 or 80. Now the family, the family size is very, very small. Okay, so even when you get older, you can't be able to depend on others. Nobody going to help you. Even nobody going to care your babies. That kind of situation is there. Okay, so that's what I'm telling. You ask a meaningful question. Okay, next slide. See here, very simple, like in the first grade, in the first grade, ஒண்ணாம் வகுப்புல படத்தை மட்டும் தான் போட்டு பாடம் சொல்லி கொடுப்பாங்க. The same thing, I am putting a simple diagram. See, this is normal, and this is mutated, right? One is normal bug, another one is mutated. It means my gene is mutated, that's what I am looking different from others. Okay, so where exactly the mutation is happening? This is the DNA, exactly this is the original. If the gene is not mutated, it looks like this. If the gene is mutated, this is the correct copy and this is the mutant copy. If the mutant copy, this is the mutant copy. Okay, so you can compare the same thing to the disease, genetic disease. Okay, next slide. Okay, so now this is the current trend is human genome and then personalized medicine. Human genome and then personalized medicine. In the sense, you take simply a drop of blood, one drop of blood from you. Okay, we can isolate the DNA of your own DNA, and then we can screen the entire genome of you. Okay, anybody knows how many genes we have, the human genes? Some approximate number. So we have, previously if you see the literature, 50,000 genes, 25,000 genes, like that. Now that is not the scenario. The number of genes we have, 19,000 genes. 19,000 genes only in our body. So the 19,000 genes, it is doing various functions. So I am going to talk about only related to some of the genes. Okay, so here, as I told you, give a one, I think the madam is here. She extensively worked for this particular human genome project, and then she can able to, if you give the blood, she can able to screen, and then she will tell you what kind of mutation you have. and what kind of disease you will prone at the age of 50 or 60. How to prevent, or postpone the onset of the disease. You can't be able to prevent. Somebody asked a question, can you able to remove all the bacteria? One faculty asked, concerned with the previous talk. It is impossible to remove all the bacteria. See, a good observer is a good scientist. You see an advertisement in the TV, like a Colgate, that you are brushing the teeth. It is eliminate all the bacteria, and then if you see carefully, one bacteria is sitting on the gum, even in the advertisement. It means it is impossible to remove the organisms. Okay, it means that is sticking in the gum. The same thing here, once you understand your genome, what you can do, yes, the DNA is mutated, I may prone to develop the disease at the age of 50 or 55 because my father had the same problem. Okay, so like that, how to postpone the onset. So that's what I discussed with the Nalathambi. Like concern with your father, mother and grandfather, definitely they are affected by diabetes. Don't take carbohydrate, avoid carbohydrate. Take less carbohydrate, more protein, more fat. Okay, so that will help you to reduce the sugar level in your blood. Okay, if you have that kind of education, see, I think those 4 people, they don't like my, I think 5, they are not liking my lecture, or volunteers, I don't know. Okay, so the personalized medicine, so that is the personalized medicine. I think anybody walked out, you can go out right now. Right? So once you understand your genome, you can postpone the onset of the disease. Consider the disease is going to appear at the age of 50. So if you follow that, yes, my gene is mutated, I am following some factors, you can postpone the onset of the disease at the age of 60 or 70. That in Bangalore, I think Nalathambi, that boss, he used a book, how to control your sugar. He is having a 300 rupees book, what to eat, what should not eat. If it's so, consider my age. My age is 75 or 60, something like that, but I am not 70, okay? So I am still mentally young, I am like a sweet 16, that is different story. So, you think the age is 70, but metabolically, the person is 60. It means 10 years he is younger than the actual age. So you have to make that kind of lifestyle. If it's so, once you understand your genome, then you change the lifestyle that will take, live longer, happily with your life. Okay, that is the personalized medicine. Even the cancer, okay, some of the drugs may be suitable for one particular person, some of them are not. Even the coronavirus, it is mutating frequently. Some people are susceptible for corona, some people are not. But that is based on the immunity. If the immunity is very less, obviously they will die. Okay, there are so many things, again, once you understand your personalized genome, that will helpful for the personalized medicine. Very recently in America, the Human Genome Project is completed long back, 25 years ago. Now they made another step. It means in 2030, within the year 2030, what we supposed to do? What we supposed to do to live longer or to live healthily? So what they did, I told you 19,000 genes. That is only in the coding region. In the non-coding region, we don't understand anything. Okay, we have to explore the non-coding regions of our genome. If you understand the coding and non-coding region, you can even you can understand the pathogenesis of the disease. Like that, then everything is the clear cut. You can understand the disease pathogenesis, and then you can go for a proper treatment. Okay, that is the human genome and personalized medicine. Next slide. Okay, please recall the 10th grade and the 12th grade book. Obviously you got higher mark in biology. This is the structure of the eye. Okay, because I'm not going to talk about any other organ, because I'm working in eye hospital. I am supposed to talk about the eye. So this is an eye. So this is cornea, this is lens, this is the anterior chamber, and this is the posterior chamber. This is retina, this is optic nerve. This optic nerve is going to the brain. Okay, so once you see the object, the light is coming through the pupil, it is in the macula, and then it is transforming the information to the brain. Yes, this is the bouquet, this is the water bottle. I can able to recognize the object. Is there any problem? Okay, so is there any problem in the cornea? It will mutate here and there. Obviously, some people lose the central vision, some people lose the peripheral vision. That is depending upon the disease. Okay, please, maybe you can interact with your parents, you can interact with your grandparents. They may have the glaucoma. Glaucoma is nothing but the eye pressure. Not only the pressure is in the heart, there is a pressure in the eye too. Once the pressure is very high, it will affect the optic nerve. So the optic nerve here, the optic nerve is gone. If it's so, they will lose the peripheral vision. They can't able to see this pillar and this pillar. They can able to see only the center. So that is one disease. If there is any problem in the retina, okay, so it will affect the central vision. They can see the periphery. They can able to see the center. Everything is is depending upon the gene or mutation. Okay, so I am going to interact with you 3 diseases. All the work done by Nidhirajan and Nalathambi. Okay, that is their PhD topic. I am going to briefly explain in a detailed manner. Next slide. Okay, see here. This is I will come one by one. That this is number 1, 2, and 3. So this is number 1. So I think you see the eyelid. The eyelid is normal. If you see some of the patient, the eyelid is like this. Is a very narrow down. See here. See the difference. This lady having a proper eyelid. This is the sister, her sister. See the eyelid. This is her son. This is normal, and again another one son for her. The eyelid is like this. So it is depending upon a particular gene. Okay. So out of the 90,000 genes, one particular gene is defective for that particular in the family. So that is inheriting to the family. Okay. So here, okay, this is the normal one, sister abnormal, and her son is normal, and another son is having the defect. So this is a genetic effect. Okay. Okay, this is number 1 disease. Another one, as I told you, glaucoma. So this is the structure of the eye. See here. So this is the iris and ciliary body. The ciliary body secretes the aqueous humor, a kind of fluid. That the fluid should go in the anterior chamber, and then this is in a kind of open angle. It will go through, and then it go through the meshwork, and then episcleral vein. That is the pathway. So you and me having a proper open angle, the angle is open, so that's what the aqueous humor is flowing in and out. So based on this flow, there is no change in the pressure of the eye. If there is any blockage in this place, what it will do, one particular gene is normal, it will go. If it is abnormal, the gene is mutated, it will form the protein interaction, it will form together, and then it will form a complex that is nothing but extracellular matrix, a kind of clump. So it will block, then the peripheral vision is gone. Okay, that is number 2. The third one, this is Nalathambi thesis, and this is Nidhirajan thesis. If you type simply aniridia, even now, simply type aniridia, A-N-I-R-I-D-A. If you type that one, I am talking about. He did PhD in 2000. This is the first picture in the world. This is his picture. Okay, so in the Wikipedia, I think this is the reference for aniridia. So we reported long back. Even some of the gene we identified in the first time in the world, we identified some of the candidate gene for eye disease. We did it. So we published in the very high impact journal that is called Nature Genetics. Okay, so I will tell you those stories later. So this is PBES, and then this is glaucoma and aniridia. I am going to discuss in detail all the 3 diseases one by one. Next slide. Okay, so this is the first one that I told you. We have the eyebrow is good, but some people having this kind of narrow eyebrow. You know why I am putting this one? This particular patient is from Huzur. They collected huge family, 5 generation family, grandfather, father, mother, like that. 5 generation. If it is in US, they will migrate here and there. They can't able to collect the samples. But in India, they are not migrating. They are sitting in one place for 5 generation. Or you collect all the samples and then you can do a excellent study. Okay, so like that, so we did all the screening. Then the name of the gene is called, if you write down FOXL2, FOXL2, that is the name of the gene. Okay, so the gene name is FOXL2. Okay, so see here, this is the structure of this particular gene. So this symposium or conference is concerned with the transcription factor related conference. So this gene is kind of transcription factor gene. Okay, so here and this is the gene structure, I think. Okay, so like recall the structure of the gene, the promoter, and then the coding regions, like that, and then it is having the ATGC start codon and start codon, and the stop codon is a TAA. Please, you interact with your faculty and they may take the class related to gene and gene structure. So this is what is exon, intron. This is the structure of the gene, promoter, and then transcript region. Next slide. Okay, so this is the how many number of, I am going to focus only 3 transcription factor genes. Please, go to next slide. Next slide. If you please note down this particular paper. Maybe I am requesting the faculties, you can give the assignment to all the students, and if they want to know, I think please go through this particular paper. It will give you all the information about the transcription factor. This is one of the excellent review. It will tell you the complete story. So what is transcription factor of the human? Okay, it will tell you all the stories. Go to the previous slide. So that next slide. See, all the information will be there. So how many what are the families? Because I don't have much time. So what are the families? What is the number? All the information will be there. Anyhow, I am picked up 3. I am going to explain only 3 of the transcription factors. Next slide. Next slide. Okay. So now the first disease related to the eyelid, that is called that is called blepharomatous Tobis epicanthus syndrome. It's a very complex name. Simply write it down. PBES, this is that's what I told you is a keywords. Okay, so Bardet-Biedl syndrome and then FOXL2, this is the name of the gene. So what we did, the functional studies were demonstrated the FOXL1 gene for PBES and reported first evidence for recessive Bardet-Biedl syndrome. Recessive in the sense, I think you please recall what is autosomal dominant, what is autosomal recessive, what is X-linked recessive, and then mitochondrial disorder. Some disease, if you see the 5 generation from grandfather to grandson, the disease, it is vertical transmission. In all the generation, they have one member is affected in all the 5 generation, at least one. If it is a recessive case, grandfather is affected, his son is not affected, but grandson is affected. It means the recessive disease skip one generation, but the severity of the disease is very high for recessive. Okay, so one is dominant and then recessive, and another one is X-linked recessive, and another one is mitochondrial disorder. The mitochondria, whatever the mitochondria, the ATP produced in our body, that is derived from mother. Whatever the mitochondria we are having, we got it from our mother, not from father at all. Some few repeats are there, that is different story. Mainly, whatever the mitochondria we have, we got it from mother. If the mitochondria is having some mutations, definitely the son or daughter, definitely they have the disease. Okay, right? So here, okay, that is the recessive. I am explaining what is dominant, recessive, and then mitochondrial disorder. Next slide. Okay, so now we will see the FOXL2 gene. What is the FOXL2? What is the beauty of the gene? See here, FOXL2. This is considered a gene. So if you see gene, the amino acid sequences. Okay, protein is nothing but yes, amino acids. So here, in the particular gene, a kind of alanine repeats. See here, you and me, if we sequence the protein sequence or the DNA sequence, there are 17 to 18 alanine repeats. That is a very interesting thing. Okay, see here, you and me having the 18 alanine repeats. See here, this is polyalanine repeat, you know, it is keep on going. Understand? Alanine, alanine, alanine. But you and me have the same 18 alanine repeats in our genome, but those that narrow down, you know, that the diseased person, if there is any one more alanine is added, or one more or two more alanine is added, is a problem. Understand? A kind of, this is a FOXL2, it is having a alanine domain, polyalanine domain. Obviously, the norm, even in the normal gene, 17 alanine are there. If there is any problem in the particular patient, if there is one more alanine or two more alanine added, definitely no, they will end with the disease. That is the message. You got it? Yes? Next slide. Okay, see here, I told you this is all done by Nalathambi. This is published in a well impact journal that is called in human genetics, human mutation, like that. The comment, you know, suppose it is very difficult to publish the paper, but once you get accepted, the reviewer, you know, they will comment, you know, they will give very good comments. If from India, you know, it is going, the paper is going to abroad for evaluation, they never appreciate this particular paper, you know, what they told you, that is called biological relevance. Whatever you are doing in the bench, that should be translated through animals and then prove. The same thing here, the comment is, it is impossible to repeat in the animal model. Even this kind of experiments, you know, it is impossible to repeat in the animal model, because all the Americans, you know, or Europeans, they are doing all the experiments in the mouse. If you go to Switzerland, if you go to other places, you know, one mouse, they will put a statue for the mouse, and then the mouse, you know, it is having some DNA on the hand. You know, because, you know, they are giving that much importance to the mouse, okay, because sacrificing all the mouse for the biological experiments. But here, in the human, you know, it is impossible to get this kind of family. Okay, this is a kind of, excellent information we are giving to the scientific community. Okay, so this is the 5 generation family. See here, this is the husband, this is the husband. Please note this pedigree. I think again I am telling the faculties, ask them to draw the pedigree, how to draw the pedigree, that kind of thing. So here, see here, this is the father. I think a person married elder daughter, sorry, the elder sister and then younger sister. He is a lucky guy. Yes? No. Are you with me? புரியுதா இல்லையா? ஒருத்தர் வந்து அக்காவையும் கல்யாணம் பண்ணிட்டாரு, தங்கச்சியும் கல்யாணம் பண்ணிட்டாரு. புரியலையா? நான் இங்கிலீஷ் பேசுறது புரியுதா புரியலையா? நான் தான் கத்தி எனக்கு நானே பேசிட்டு இருக்கேனா? So this person is married the elder sister and the younger sister. The elder sister having 3 sons. The second son is affected. He is the second son. His first son is normal. She is the normal mother. But the sister, you know, the sister, she is having some maternal issue. Even, you know, she can't able to become pregnant because of the defect, you know, she is infertile. Okay, we proved, you know, because of the mutation, because of the FOXL2 gene mutation, she is impotent, and then, you know, she can't able to have the baby. Okay, so here, I think, you know, this boy, you know, having this kind of same variation like her. Okay, that is because the gene is, sometimes it is expressed, sometimes it may not expressed. So the same way here in this family, this kind of, phenotype is, appeared. So I think we proved in a perfect way, in everything, like sequencing, gene expression studies, everything we did it. So this is the point number 1. Understand? That is FOXL2. Next slide. Next slide. Okay, so now we will come to the second disease, the glaucoma. Glaucoma, that is it is secreting in the aqueous humor, and then the aqueous humor is going through the meshwork. If there is any blockage, they will lose the peripheral vision. I told you, right? So the gene name is called FOC SIX6. This is primary open angle glaucoma. The primary open angle glaucoma. The name of the gene is called SIX6. Again, this is the transcription factor gene. Please understand, I am picking only the transcription factor genes for this conference. So here, I think this is the name, this is the gene, and we identified the mutation in different regions of the gene. Next slide. And see here, in the promoter region, not in the coding region, this is the promoter region. Promoter region is very, very important, where the transcription factor binds. Okay, so here, see here, these are the transcription factors, and we identified a 4 base pair deletion. In the previous one, there is 1 more or 2 more alanine added, but here deleted. It means a DNA, sometimes it may add, sometimes it may be delete. So here I given an example, addition in the previous one, here it is a deletion. So there is a 4 base pair deletion. Because of this, there is a defect. Next slide. So you can prove in a GFRA-FISH model. Whatever you are doing in the experiment, you have to prove that is called a biological relevance. You can prove it in the GFRA-FISH model. So this is the way to do it. So you can take the SIX gene and then you can delete the 4 base pair, make a plasmid, and then put it in the vector. You take that one and then put in the GFRA-FISH egg. I think I am going to ask you one question. The fertilization, what is fertilization? For fish, it is a external fertilization or internal fertilization? External, correct? So the male and female, and then the female, it will release the egg, and then the male, release the sperm. The fertilization occurs in the water, right? So you can take the egg, the fresh egg, and then you take that plasmid, with containing that the 4 base pair deletion, and then you inject that DNA into the egg. Okay, then you put it and then fuse together after the fertilization. Once the sperm is entered, that is fertilized within a short span of time. Next slide. So why we have selected the GFRA-FISH? The GFRA-FISH is very, very transparent. You can see in the naked eye, even in the globe, because we are working on the eye, the globe size, the globe in the entire eye, suppose consider this is the shape of the eye, this much. But once the mutation happened, 4 base pair deletion, the globe size is very low. You can compare the globes, because he is working in the previous speaker, he is from Korea. So you like that, you have to connect it. The ஒரு ஜோக் கூட இருக்குல்ல, ஒரு ஸ்டாம்பை கொடுத்தா அவன் ஏதோ ஒரே ஒரு ரூபாய்க்கு தான் ஸ்டாம்பு கொடுத்தான், அவன் இங்கிலாந்து ராணி பற்றி பேசுகிறான். இது ஒரு ஜோக் இருக்குல்ல, சரத்குமார் அண்டு தட் வடிவேல், ரைட்? ஏன்னு ஒரு படம் இருக்குல்ல, அது பார்த்தீங்களா? போய் பாருங்க நீங்கள். Okay, so even a single thing, you have to connect it, built up many things. For that you need more information. For that you have to read in deep. Okay, so I am comparing the globe of the Asians, especially in Indians and Koreans, because the globe size is very small when compared to us. Okay, the same way here, if there is any 4 base pair deletion, the GFRA-FISH, see here. So this is the I think we have to turn off the light. See here, the globe, okay, so this is the normal fish, the globe is big and the globe is reduced here. This is the mutant, 4 base pair deletion. You compare this globe and this globe. Okay, so like that, if you go inside, if you go to the inside of the retina, and what are the structure is reduced. Okay, I don't want to go in detail what are the layers and everything, the flexiform layers and everything. So that is completely reduced. It means that transcription factor SIX6 playing a major role for glaucoma. That is the conclusion. All right, next slide. So next, the aniridia, Again, that is the name of the disease. Here, next slide. So here the gene is PAX6. So I told you 3 genes. One is FOXL2, another one is the SAX6, the third one is the PAX6. All the 3 are transcription factors. Okay, so that is relevant to the conference. So again, here this is the gene, and then what Nidhirajan did. So those, please, go back to the future. Okay, 2000, in the year 2000, just completed the human genome. There is no sequencer in India. No sequencer. So what we did, we all three, what we'll do, either we'll do sequencing ourselves, manual sequencing, or SSCP analysis, or we'll send the sample to Korea, Switzerland, and then we'll do the sequencing. Okay, no sequencer in India at all. Even that day, those days, we did the human genome sequencing in our lab. Okay, then now we have high-end machines. We have 4 sequencer, we have MySeq, so many sequences we have in our lab. Those days, with a limited source, but you have to thinking small is crime. You have to think big. Those days, we thought big, and then that's what he published more than 4 or 5 papers all together. Okay, so again this is the PAX6. Next slide. Okay, so if you are really interested, again, these are the publication of Nidhirajan. You can see here are the 3 papers. See here, Nidhirajan, Nalathambi, myself. I'm the guide, so obviously I'm the corresponding author. So all the 3 papers. So here, what emphasize, nobody reported those days what is the genetic variation for aniridia. Again, aniridia, you can ask a question. Somebody told you, sir, you told about narrow eyelid, and then you told about glaucoma. What is aniridia? So aniridia in the sense, iris. Iris is a part of the lens. If there is any mutation in the PAX6, this is a complete iris. It is like a mild, moderate, severe. Sometimes full iris, sometimes partial, sometimes only the rudiment. But see, you are normal. That's what you don't know the suffering of the patients. In America, they have an association. All the 3 diseases, aniridia, they have an association. Now we interacted and tried to get some fund. Okay, aniridia association. Because the parents, they have to give some good, positive words to their children, because they have aniridia. Okay, they formed a group and then they discuss, is there any improvement? So where is the treatment? Okay, we are getting lot of emails. Once you get published this paper, they will write, I think my son is having problem, what to do, how to avoid this, I have to understand my sequence. Because right now people have money, but they are not taking care of their health. Again, I'm telling you, I'm giving a message. If you want to live healthily, happily, please, understand your own genome, change your lifestyle, live longer. That is the message. Next slide. Okay, I think maybe you guys are mentally tired, because I'm giving more information. Sometimes, for some people, whatever I'm telling, it will go into your ear, it will stick on your brain. For many people, it will flying overheads. Okay, it is flying overheads. Okay, let him talk, Let him talk. It is flying overheads. Okay, I will conclude like this. See, Nidhirajan, and Nalathambi, long back in the year 2000, before Dr. Abdul Kalam became a president, he was the scientific advisor to the Prime Minister, Vajpayee. Okay, that time he visited our lab. We established our lab. So I think, see here, Dr. Abdul Kalam, the Nidhirajan, head of the department, Nidhirajan. See the young, handsome guy here. Who is this? Me. Hey, you can't able to believe me. I have to dye my hair, This is me. Next slide. And every Thursday we have a lab meeting. Please, at least, there are a lot of trees in our garden, in our campus. Form a group, eat lunch together, discuss something, related to subject. Don't gossip, don't, rubbish, don't be rubbish. Okay, discuss something, please. Okay, every Thursday we have a meeting. See, this is again Nidhirajan, and this is me, and this is a very well-known immunologist in India, Dr. Muthukarpan. See, again, there is a lady section and gen section, we put it that way. Next slide. And Nalathambi is, Nalathambi is here. This is Nalathambi. And this is Nidhirajan. Even, our lab, see, I am collaborating with many countries. Okay? Except Africa, I visited all the countries, all the labs I know. I given many talks in more than 20 or 30 universities in US. So I think this is he is the director of SERI, Singapore Eye Research Institute. We went to many conferences to Singapore. So he is working on myopia. What is myopia? Why people are wearing the specs? This is a progressive, because aging. I'm reading glass. Okay, it's a myopia. So he is working on myopia. If there is any near work, obviously problem in the eye. They have to put them in thick lens. Okay, so the Singapore, they are pouring a lot of money for myopia research. Okay, so he visited our lab those days. I think this is the kind of scientific interaction we had. That's I'm selecting. I have millions of pictures in my laptop. I think I picked up very few. Okay. Next slide. Okay, I think as I told you, I have more than 25 PhD students. So I think these are my PhD students already graduated. Most of them are settled in the US. I think Nidhirajan and Nalathambi is here because to teach you well. That's what they are here. They have sacrificed their life for you. Yes or no? So read, please follow his instructions, read well. Okay, these are my PhD students. Next slide. Next slide. Thambi, okay, I think I got many awards. Even I got the 2 awards from this is the award from President of India. I went to the Rashtrapati Bhavan. I got the huge award. Okay, and this is Tamil Nadu Scientist Award, and another 2 awards from the Health Minister. That time the Congress government, Ghulam Nabi Azad was the Health Minister. I got those awards, and Dr. Abdul Kalam, when he was the President, a farmer, and after that he inaugurated our institution, AMRF. That time he came and interacted with us. This kind of interaction. What he asked me, sir, right now the human genome project is completed. People are asking my genome. The cloning strategy. The cloning is, I think that is another strategy. The cloning is not, ethical. Here I think, suppose story that, you have seen the Jurassic Park, the movie, Jurassic Park. They clone the, yes or no, Jurassic Park. So Jurassic Park, those days, the mosquito got the blood from the dinosaur, and then it has become fossil. Okay, they have taken the blood, isolated the DNA, and then they have taken the DNA, and then they put it in the frog egg. Okay, like that, they developed the dinosaur. I'm talking about that book and then story. Okay, they can able to produce only the female dinosaur, not the male dinosaur. English padamum parunga thambi, Tamil padam paakadinga, English padam parunga. Okay, okay, so like that, you know, he asked what kind of can I able to give my DNA to them. That kind of discussion is going on with the president, former president. Next slide. I think this is my current students. Like they left long back, still I have students, they are working for us. Okay, I think I can stop here. If there is any question, I will ready to answer. Next slide. Thank you. இது என்னது? இதை தான் சொல்லிட்டு நான் இல்லையா? Yes. So what is this? Yes. It means you can because you think you can. என்னால முடியும் அப்படின்னா முடியும். Today is not the end of the world. இன்னைக்கே வந்து கூட அழிஞ்சிர போதா? அழியாது. It will take long time to ஒரு காலத்துல இன்னும் ஒரு கோடி கணக்கான ஆண்டுகளுக்கு பின்னாடி சூரியனே இருக்காது. அது வரைக்கும் நாம இருக்க மாட்டோம். That is different story. Okay, so today is not the end of the world. Please, you can because you think can. Okay, I will stop here. If you have any question, I already told you, write in the question to the notebook. If you have any question, I am ready to answer. Thank you.
Hello, check.
Thank you, sir. Thank you so much. Thank you for that wonderful and interesting speech. Students, I want you to settle down immediately. Kindly please settle down. You will be served lunch. Please settle down. I know you're hungry. It is past our lunch time. I know that. But please settle down. Only after the session, we'll be going for lunch. Please settle down. We have questions and we need to get it answered. So I want you to settle down. Yes, now the session is open for discussion. Yes, ma'am, you have a question? Hello, sir.
Thambi, I think he is asking some question. நீங்க தான் தம்பி கொஸ்டின் கேக்குறீங்க. கொஸ்டின் கேக்குறீங்களா போறீங்களா?
Hello, sir. So, FOXLA gene. You said about FOXLA gene.
Yeah.
So, there is a probability in the first trimester the mother will be tested for.
Okay. Super.
The mother will be tested for syndromes, especially chromosome number 21, Down syndrome and all can be checked. Is there a possibility that we can check FOXLA gene also?
Hello.
Yeah. Okay, sorry. Okay. Talking about the prenatal diagnosis.
Yes, correct.
அப்படின்னா என்ன தெரியுமா? ஒரு அப்பா நார்மலாக இருக்காங்க, அம்மா நார்மலாக இருக்காங்க, ஆனால் பிறந்த குழந்தை வந்து டிஃபெக்டிவாக இருக்குது. இப்போ என்ன பண்ணுவாங்க, ரெண்டாவது குழந்தைங்க நார்மலாக பிறக்கணும் அப்படின்ட்டு ஒரு கியூரியாசிட்டி இருக்கும், எஸ்பெஷலி மாமியாருக்கு இருக்கும் இல்லை? எஸ் சார், நோ? So in that case, you know what we supposed to do, we have to sequence the father's DNA, mother's DNA, and then see the exact where the mutation is, and the mutation வந்து the affected child வந்திருக்கா அப்படின்னு பார்க்கணும். Okay, so now 3 pictures, father, mother and then affected child. இப்போ இவங்க என்ன கேட்குறாங்கன்னா, this is sperm, this is egg fused together, form a zygote. புரியுதா? once zygote ஆயிடுச்சுன்னா அவ்வளோதான். ஓகே, ஒன்றும் பண்ண முடியாது. ஓகே, அதை எப்படி டெஸ்ட் பண்ணலான்னா, once they conceived, okay, conceived or 4 weeks, okay, amniotic fluid, okay, amniotic fluid. இப்போ நம்ம எங்கள் ஹாஸ்பிட்டல் எடுக்க மாட்டாங்க, சென்னையில் போய் you supposed to take the amniotic fluid in a small volume, that is called, நிறைய படிங்க தம்பி, நான் ஒரு வார்த்தை சொன்னால் நீங்கள் ஒரு புக்கில் இன்ஃபர்மேஷன் இருக்குது, that is called liquid biopsy. That the liquid biopsy, know from that, you know, you can take the DNA and then அவங்களுக்கு ஆன்சருக்கு சொல்லிடலாம். ஓகே, இந்த குழந்தை, இந்த கன்சீவ் ஆன குழந்தை மறுபடியும் வந்து டிஃபெக்டிவாக பிறக்க போதா நார்மலாக பிறக்க போதானுங்கிறத நம்ம சொல்லிடலாம். ஆனால் அந்த ஃபாதர் அண்ட் மதர் தே ஹாவ் டு டிசைட் தெம்செல்ஃப், அபார்ட் பண்ணலாமா. சப்போஸ் அது மறுபடியும் அதே டிஃபெக்டாக இருந்துன்னு வச்சுங்க, அந்த அப்பா அம்மா தான் டிசைட் பண்ணோம் அபார்ட் பண்ணலாமா வேணாமான்னு. But the scientist, what they will do, this is the scenario for the baby. அப்படின்னு சொல்லலாம். ட்ரைசோமின்னு ஒன்று இருக்குது மேடம். சி இஸ் டாக்கிங் அபவுட் த குரோமோசோமல் அபரேஷன்ஸ். நம்ம ஹியூமன் குரோமோசோம் எத்தனை தெரிஞ்சவங்க மட்டும் சத்தம் போடாதீங்க, கை தூக்குங்க பார்க்கலாம். ஹியூமன் குரோமோசோம்ஸ். சூப்பர், எக்ஸலன்ட். உங்கள் பேர் என்னம்மா? திவ்யா. சி இஸ் த ரைட் ஆன்சர். 23 pairs. 23 pairs. உட்காருமா. 23 pairs, 22 autosomes and then XY chromosome. 22 autosomes, you XX, I am XY. That's what I am male, you are female. Okay, so totally 46 chromosomes there. சரியா? அப்புறம் உடனே மைட்டாசிஸ் வாங்க. மைட்டாசிஸ், when the cell divide, equal number 46. if it is a மைட்டாசிஸ் தான் சொன்னேன். so மியாசிஸ்ல எப்படி இருக்கும்? the n number is 1n. 2n for mitosis, n for meiosis. அப்பனா 23 தான் இருக்கும். எக்ல 23, ஸ்பெர்ம்ல 23, ரெண்டும் ஃபியூஸ் பண்ணா நார்மல் சைகோட். sometimes that is called chromosomal aberrations. chromosomal autogamy அது சொல்றாங்க இல்லையா? so சில சமயத்துல chromosome நம்பர் 1 வந்து 2 இருக்கும், diploid இருக்கும், அது like that. if it's so, the trisomy better. Trisomy. so if it is a trisomy, obviously the baby having some problem. okay, the baby born with some defect or syndrome, not only problem in the eye. இப்போ இவர் நீதிராஜன் ஒர்க் பண்ணார் இல்லையா, அந்த அனரீடியாவிலே 3 டைப் இருக்கு. ஒன்று கண்ணில் அஃபெக்ட் பண்ணும், ஒன்று கிட்னியில் டியூமர் வரும். சில குழந்தைங்களுக்கு ஐலையும் ப்ராப்ளம் இருக்கும், கிட்னிலையும் டியூமர் இருக்கும். சில குழந்தைங்க ஐல ப்ராப்ளம் இருக்கும், மென்டல் ரிட்டார்டேஷன். everything I think is a problem in the gene or the chromosomal aberrations. okay, thank you Divya, at least you asked the right answer, a right question. யாருங்க உங்களுக்கு வாத்தியார்? ஒரு 5 மார்க் அதிகமாக போடுங்க இந்த செமஸ்டர்ல. oh I see, you are Ferrier. I think I know the zoology department. what is her name? okay, I think she did PhD in with Gunasekharan. okay, zoology நீங்க? சரி. yeah.
So what kind of method you used to inject the zebrafish model gene injection?
okay, okay, it's called micro injections. இப்போ நீங்க நமக்கு ஊசி போடுறீங்க இல்லையா, அது அதனை எத்தனை காஜ் நீடில் தெரியுமா? அதெல்லாம் தயவு செஞ்சு நீங்கள் மிக்சர் சாப்பிட்றீங்களா, மிக்சர் அது பேப்பர் தானே கட்டித்தரான், சாப்பிடுங்க. அந்த தூக்கி போடுறதுக்கு முன்னாடி அந்த பேப்பரில் என்ன எழுதியிருக்கான்னு சொல்லிட்டு தூக்கி போடுங்க. த சேம் வே தட் காஜ் நீடில் 23 காஜ் நீடில் ஆர் 25 காஜ் நீடில், that is nothing but the டக் இருக்கலாமா, அந்த டயாமீட்டர் உங்களுக்கு தெரியணும். ஓகே, வித்தவுட் அஃபெக்டிங் தட் பர்டிகுலர். அதுக்கு வந்து அமெரிக்காவில் இருக்கும்போது ஐ ஒர்க் வித் பிரெயின், அதெல்லாம் பெரிய பெரிய ஸ்டோரி. நீங்கள் என் பேரை மட்டும் அடிங்க, பப்மெண்ட் தெரியுங்களா? பப்மெண்ட்டில் என் பேரை அடிங்க. ஸோ லைக் வி ஒர்க் வித் பிரெயின் டியூமர். அந்த பிரெயின் டியூமரை க்யூர் பண்ணுறதுக்கு ஒரு கேன்சர், அது வைரஸ் வந்து உள்ள தள்ளணும், அந்த பிரெயின் டியூமரில் உள்ள தள்ளணும். அப்படிங்கும் போது இந்த மாதிரி நீடில் தான் வி யூஸ்ட். ஸோ தட் இஸ் கால்ட் மைக்ரோ இன்ஜெக்ஷன். கன்சிடர் திஸ் இஸ் அ ஃபிஷ் எக் அண்ட் தென் யூ சப்போஸ் டு புஷ் தட் த பிளாஸ்மிட் இன் டு த எக் இல்லையா? அந்த பிளாஸ்மிட் வந்து அஃபெக்ட் ஆகக்கூடாது. அதுக்கு தகுந்த மாதிரி அந்த டயாமீட்டரை வச்சு நீங்கள் வித்தவுட் டேமேஜிங் த எக் புஷ் இட் இன்சைட். யூடியூப் பாருங்களாங்க, கண்ட கருமமும் அந்த இதில் யூடியூப்பில் பார்க்குறதுக்கு சயின்டிஃபிக்காக ஏதாவது பாருங்கள். ஆல் இஸ் தேர். ஓகே, இல்லை she is asking, see there is a candidate gene. One, if it is a candidate gene in the sense, this particular gene, if it is mutated, it will form the small eye. That is called candidate gene. That is some gene for breast cancer, someone for this one eye disease, like that. One particular gene that is called CCT, that is name of the gene, very recently identified. You take that gene, you make a mutation, and then you take the mutant, push it to the egg, and you grow the egg. Then that the fish, it will defect in the eye, globe size.
Sir, any ethical clearance?
Yeah. Very good. இல்லை இதெல்லாம் வந்து இவங்க கேட்குற கேள்விலாம் இட்ஸ் அ வெரி வேலிட் கொஸ்டின். இப்போ ஹலோ தம்பி, கொஞ்சம் பேசாமல் தம்பி ப்ளீஸ், நான் கொஞ்சம் பேசுகிறேன், அப்புறம் நீங்கள் இப்போ பேசுங்க ப்ளீஸ் தம்பி, கொஞ்சம் அப்புறம் பேசுங்க தம்பி ப்ளீஸ். அதாவது ஐ திங்க் சி இஸ் ஆஸ்கிங் an excellent question. ப்ளீஸ் உட்காருங்க. வாட் இஸ் எத்திக்ஸ்? எத்திக்ஸ்னா என்னன்னு தெரியுமா? நான் பாட்டுக்கு சும்மா இப்போ மவுஸெல்லாம் கொள்றோம்ல, இப்போ இவங்க கேட்ட கேள்வி அருமையான கேள்வி. இப்போ நான் அதில் நிறைய வேலை பார்த்துருக்கேன். கன்சிடர் த மவுஸ். ஒரு டியூமர் வருதா வழியானு இப்போ பண்ணுறதுக்காக யூ டேக் த டியூமர், பிரஸ்ட் கேன்சர் செல்ஸ் ஆர் யூட்டிரஸில் நிறைய கேன்சர் வரும், யூரினரி பிளாடர், ப்ராஸ்டேட் கேன்சர். அந்த செல்லை வந்து இன்விட்ரோவில் அந்த செல்ஸை எடுத்து நியூட் மவுஸ், இம்யூனாலஜி நியூட் மவுஸ்னால் அந்த அந்த டியூமர் செல்ஸை வந்து இங்கே இன்ஜெக்ட் பண்ணுவாங்க. வித்தின் 15 days it will form the tumor. அந்த டியூமர் வந்து ஒரு பர்டிகுலர் சைஸ்க்கு தான் இருக்கணும், அதுக்குள்ளே நீங்கள் அந்த அனிமலை கொன்னுடணும். ஓகே, நான் எதுக்கு சொல்கிறேன்னா எத்திக்ஸ். அந்த 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 ஏற்கனவே அந்த அனிமலை நீங்கள் வந்து இது பண்ணுறீங்க, தொந்தரவு பண்ணுறீங்க. அந்த டியூமர் பேர்டன் இருக்குல்ல, சில சமயத்தில் அப்படியே அதை தூக்கிட்டு நடக்க முடியாது, அதனால் சாக்ரிஃபைஸ் பண்ணிடுன்னு சொல்லுவாங்க. அதனால் மவுஸுக்கே இப்படி பண்ணும்போது ஹியூமனுக்கு எத்திக்கல் கமிட்டி வாங்கணும். நீங்கள் ஒரு கிராண்ட் எழுதுறீங்க, ரிசர்ச் கிராண்ட், ICMR ஆஸ்கணும் இந்த எத்திக்கல் கிளியரன்ஸ். இது எது நீங்கள் செல் கல்ச்சர் பண்ணுறீங்களா, ஹியூமனை டச் பண்ணுறீங்களா, DNA எடுக்கிறீங்களா, வெதர் இட் இஸ் இன்வேசிவ், அங்கே இன்ஜெக்ட் பண்ணுறீங்களா, யூ ஹேவ் டு ஆர் யூ கேன் கெட் த கன்சர்ன் ஃப்ரம் த பேஷண்ட். நான் ஒத்துக்கிறேங்க, எல்லாம் நீங்கள் என்ன பண்ணுறீங்களோ ஒத்துக்கிறேன்னு சொல்லி கையெழுத்து வாங்கிட்டு தான் பண்ணணும். We have an ethical committee, we have proper channel to do the research. Very good question. Yeah, I think very recently they modified. See, I am in many committees in government. Okay, so she is asking. Previously, again we can classify the animals: small animals, big animals. In America, they are using monkey, pig, dog, goat. Okay, so here people are using only mouse or zebrafish. Previously, there is no clearance for zebrafish. Now the DST or the DBT, they told definitely, even at least you had to get the consent from the committee. Yeah. சரி, அவ்வளோதானே. தேங்க்யூ. ஓகே, ஐ திங்க் வாட் ஷி இஸ் டெல்லிங், அது ஒரு திருக்குறள், திருவள்ளுவரோட முடிச்சிருவோம். அதாவது செவிக்கு, நான் வந்து சாப்பிடாம 2 நாள் பேசுவேன். ஓகே ரைட். சரி, இப்போ திருவள்ளுவர் குறள் சொல்லியிருக்காருல, என்ன சொல்லியிருக்காரு? செவிக்கு உணவு இல்லாத பொழுது சிறிது வயிற்றுக்கு ஈயப்படும். நான் உங்களுக்கு நிறைய சொல்லி கொடுத்துட்டு இருக்கேன். பசிக்குமா? பசிக்க கூடாது. ஓகே, ஸோ பசிக்காதவங்க இங்கே இருங்க, பசிக்கிறவங்க போய் சாப்பிடுங்க. தேங்க்யூ.
Students, please settle down. We have not broken for lunch. Please settle down. Sir, thank you so much for your valuable time, and I hope in 2 days we'll be having enough time to
Yeah, sure. Okay.
Thank you so much. Lunch is served in the BCA block. I hope everyone will be knowing the BCA block. Yes, for the newcomers,
Hello.
we'll take you. Please come us and join us for our lunch, which will be served in the BCA block. The time now is 12:40. So we'll be coming back again at 1:30 sharp. Okay, 1:30 sharp. I want everyone, biotech students, please stay here. I want you to stay here. Thank you so much. Biotech students, I want you to stay here. Please rest of us, please join us for lunch. Biotech students, I have an announcement for you. I said biotech, if I'm meaning first BSc A and B, second BSc A and B, third BSc A and B, and first MSc and second MSc. Please stay back.